Okay, pretty wild, huh? How many of you guys had the opportunity to dive on a coral reef? Isn't it amazing? I mean, the number of fishes is just like, wow, it's astounding, you know? And there's a lot of cool stuff about coral reefs and interactions and symbiosis and the importance of certain fishes. But a lot of coral reefs are being degraded as a consequence of overfishing, as a consequence of destruction to the reefs, and now more recently as a consequence of climate change, where warming waters causes bleaching by causing the zooanthellae, which are the symbiotic organisms that are within the, let's see, those guys are, corals are in the same family as hydras, same group as hydras. And those are called what? You guys remember? Hydras and corals. It's not prolifera, it's sponges. Come on, guys, didn't you have zoology? Help me out here. What are they called? What are that? The corals and hydras are in the which group now? Hydrozoans, are they? Okay. Yeah, hydrozoans. And the zooanthellae are the symbiotic organisms within those. Okay, thank you. All right, at any rate, so what these guys did was something pretty cool, and that was that they looked at marine protected areas versus non-marine protected areas, and the former contained healthy coral reefs, and the latter had been damaged or what have you, and they were dominated by algae, right? And then they showed that the larvae of corals, which are called planula, and the reef fishes preferred water from the marine protected areas. So what do I mean by that? And I think, did I give you guys this? Maybe not this. I didn't give you that, did I? Well, no. Okay, we'll talk. But at any rate, this just shows an example now. They took water from marine protected areas versus non-marine protected areas. They had 3 different species of coral, and they showed that the percent time that the little planula, these are the larval corals as they're floating around, the percent time they spent in these different waters, right? And they liked the marine protected water better, and they did an analysis to show what was going on. They also looked at different cues in the water. They looked at the different compounds that were in the water, either algae or coral compounds. Okay, so bottom line, larval corals like water that comes from coral reefs better than from reefs or areas dominated by algae. Then they looked at fishes. These are typical families of fishes, so your dark lines represent families of fishes on one side, right? And these are typical fishes that live in coral reef regions. And they did the same thing. They captured these fish from marine protected areas. Of course, these fish also live in non-marine protected areas because it's maybe some of the only places they can live. And then they showed that they prefer the water from coral reefs, shown in color, in your case kind of shaded, versus water from non-marine protected areas, right? So the fish also like water from coral reefs more than they like water from algal areas, okay? Then they did something really cool.
Is there something possibly the filtration of the coral may be doing to the water?
It wasn't the filtration, it's more the chemical cues that they're giving off. So then what they did was they took water from marine protected areas in 2 ways, right? And they filtered the water, but then they added back in coral reef, the essence of coral reef. I can't remember what it is. And then they filtered back in the essence of algae, right? So now they took, because maybe it's something in the water they don't know what it is, right? And so it's a nice experiment. So they took water from coral reefs, filtered it, and then put coral cues in the water, in that water, and then put algae cues in that water. And the fish liked the water where there was coral cues over algae cues. So it wasn't the water just because it came from the coral reef area, right? Does that make sense? What's that? Something, right? I got to read the paper again. They mention what it is. Then they did the same thing with water from non-marine protected areas and water from non-marine protected areas with coral chemical cues. So now that's shown in white. And so wherever, basically, the bottom line is, wherever there were coral chemical cues, both the larvae of the coral and the fish were more attracted to that water than they were to water from algae-dominated areas, right? So what does that mean? Why is that important? Well, it's interesting from the standpoint of the ecology of these fishes, but perhaps what's even more important from the standpoint of management is that that take-home message means that those animals are going to seek out high-quality habitat as opposed to degraded habitat. And so it's going to be much harder to recover degraded habitat if the fishes and the corals themselves aren't attracted to it. Right? So how do you get them to come to an area to form a reef? Because remember, you have to have corals to form the reef, but the fishes are performing essential functions, ecological functions on that reef. And one of the things these fishes do is they keep algae down. They eat the algae and whatnot, right? And that helps keep the coral healthy, right? So interesting study, and that's a really cool study and a really nice experiment. So it just shows how you can apply science to, you know, sort of a real world or management situation and what that might mean for difficulties in recovering coral reefs. Okay, and why are coral reefs important to humans? Any ideas? Besides ecotourism? No, I don't know that they absorb CO2, but they certainly create a lot of biomass, that's for sure. What's that? Well, a lot of communities around all the world, right, fish on coral reefs. And so a lot of food comes off of coral reefs for people all over the world. And so when you have degraded coral reefs, you're going to have less food available for people. Right? So it's important in a lot of ways. Okay, so now today we're going to talk about primarily living in water, but you know we're going to kind of contrast that with life in water versus life in air, but we'll focus on living in water, as this is the last lecture where we're discussing, you know, sort of fishes, if you will. And these are the kinds of things that we're going to discuss today. We'll talk about differences in O2 content, temperature and pressure, buoyancy versus gravity, movement in water. We've talked about that to some degree. We'll spend a fair amount of time talking about osmoregulation, and then we'll also get into sensory systems. So these are the topics we're going to cover, okay? And we'll do them each one. So you don't necessarily have to write them all down, but you will write them individually as we go. So the problem of living in, I'm going to do this, the problem of living in water is that the O2 concentration of water is much, much lower than the O2 concentration of air. So you have a problem of extracting, you know, oxygen, which you need in order to have your cells metabolize when oxygen concentration is really low. And on top of that, oxygen concentration, noted by here, if you guys all remember your chemistry, right? It varies inversely with temperature and salinity. So as temperature goes up and salinity goes up, oxygen concentration goes down. Okay, so now the way fishes have dealt with this is kind of a neat mechanism. Okay, first off, obviously they're using their gills, and their gills basically allow capillaries of their blood vessels, right, and the blood flowing through those capillaries to be in direct contact with water. Okay, and so the water flows into the fish through the mouth and out the gills in one direction. And this enables the fish to set up something called a countercurrent exchanger in the gills. And then we're going to explain what that is, countercurrent exchange mechanism. And that's a really important mechanism, and it's based on the physics and chemistry of concentration gradients. So we'll get into that in a moment. But yet some fish cannot meet their oxygen needs using gills alone, and some of them breathe, and they actually gulp air, and then they either have sort of kind of like an adjacent swim bladder that enables them to extract oxygen, or they actually can extract oxygen out of their buccal cavity. And this usually occurs in fishes that live in very oxygen-poor environments. But now let's talk about this countercurrent exchange mechanism. And I believe, did I give you that figure? I should have, right? Okay, so as we mentioned, it's figure 4.1. Is that in your list? Yeah? It's not? Let me see. That's 4.2. Okay, well then this is something you can write down. So as fish, and this is 4.1 in your book, so the one-way direction of flow, right? Water flows in the fish's mouth and across the gills. You all know that. Here's another another illustration of it. Water flows in and then across the gills, okay? When it does that, when the water flows in one direction, noted by these arrows right here, these are the lamellae. These are the individual components of the gills themselves, right? They're like the filaments of the gill. The blood flows in the opposite direction of the water. Okay? That's called the countercurrent mechanism. Water flows one way, blood flows the other way. Okay? And why is that important? Because this sets up a unique concentration gradient that allows the fishes to extract more oxygen out of the water than if the water and the blood were flowing in the same direction. Okay? So what do I mean by that? This is what happens in a countercurrent exchange mechanism. Two things to recall. When you have a concentration gradient, right, any concentration is going to be dependent on the direction, well, I should say, the place, a molecule of where it's highly concentrated, it's going to move to where it's low in concentration, right? Remember kind of osmosis, movement of molecules, right? You go from a place of high concentration to a place of low concentration. So that's the direction of O2 concentration. And then the flow of that molecule is going to be dependent on the magnitude of the concentration gradient. So if you have concentration gradients that are relatively similar to one another, you're going to have a little bit of flow from high to low concentration. But if you have a concentration gradient that's very different from one another, you're going to have lots of flow over a short period of time. Does that make sense? Okay? So now let's look at what happens in a countercurrent mechanism versus a mechanism where the water and blood are flowing in the same direction, and we're going to explain that one first. So here's a situation where water and blood are flowing in the same direction, and this is on the y-axis the percent saturation of oxygen in either one of these two compounds. So initially, if you have water coming into the gills, and if blood was flowing in the same direction, so obviously it's deoxygenated, right? You're trying to oxygenate that blood. It's going to have a really low O2 concentration. So the magnitude of the difference in concentration gradient is going to be great. Right? And so consequently, oxygen is going to want to flow in a lot from water to blood, right? And so what happens is that as that oxygen dumps into the blood, the concentration equilibrates at, say, around 50% oxygen saturation in the blood. So you're only able to extract 50% of the O2 out of the water and get it into your blood when, or if, because it doesn't happen this way, if blood and water were flowing in the same direction. Are you with me? Okay, so now when blood and water are flowing in the opposite directions, as is depicted here, so here's the in-current blood, excurrent blood, so that means the blood that's going into the gills, blood that's leaving the gills, and then here water is coming into the gills from the opposite direction. So these are all relative sort of terms. So this means high O2 concentration in the water, low O2 concentration in the water, low O2 concentration in the blood, high O2 concentration in the blood. When you set up a countercurrent exchange mechanism, the thing that happens is all along the lamellae, the concentration of oxygen in the water is slightly higher than the concentration of oxygen in the blood. So that means oxygen is always flowing from water to blood. Okay? But it's a small level of concentration difference. But that small level of difference enables the fish to extract the most oxygen out of the water, because all along that gradient you're going to have a slightly lower concentration of oxygen in the blood than you do in the water, and eventually when that blood leaves the gills, it's going to be around, somewhere between like 80 or 90% saturated rather than only 50% saturated, because all along the length oxygen is being poured in, if you will, into the blood, right? And that results in extracting more oxygen from the water than you would if you had them flowing in the same direction. Does that make sense? Okay, that's called a countercurrent exchange mechanism. It not only happens with oxygen concentrations, it also happens with heat, it also happens with concentrations of other molecules, and etc. That physical process of allowing the concentration gradient to always be slightly greater in one place versus another enables this to occur. Okay? Important thing to understand. So that's pretty cool. Okay, so now let's look at temperature regulation. So even though oxygen is really low in water relative to air, water conducts heat 24 times that of air. Its heat capacity, that is how much heat can it absorb, is 3,500 times higher. So that means you can lose heat while you're in water. And think about it when when you've swam in cold water, right? Remember when you're sitting there, okay, and you're hanging out, and you're in your pool, and you're like, oh, I hope nobody sees me do this, but I gotta pee. And you pee, and that warm pee goes all over through your trunks, and you're like, oh, that feels so good, And then all of a sudden, that wisp of cold water comes by, and all that warm pee goes away, right? And you're like, oh, that's cold, right? Right? You've all experienced that. How many of you have peed in a pond? Come on. Oh, come on. Yeah, there. Dylan's honest. Okay, now think about it on a much smaller scale, like the boundary layer right next to your skin. You're warming up that water right next to your skin, right? And then that heat is being carried away. Now, it could be carried away when the water flows through, but the other way it's carried away is that the water is just denser. And so that molecular movement, which is what heat really is, just gets transferred from one water molecule to the next. And so the heat next to your body basically gets transferred into all those water molecules, and you lose heat, right? That's the principle of a wetsuit. In a wetsuit, you have that neoprene layer. There's a layer of water between the suit and your skin, and you heat up that layer of water that's in between there, and that's what keeps you warm. That's why a wetsuit keeps you warmer than if you didn't have a wetsuit. Okay, make sense? Okay, so since water can conduct heat, there's problems in maintaining heat when you live in water, and there's a couple ways that fishes do this. First is this idea of regional heterothermy, that parts of the body can be warmer than others, right? And that's a consequence of a countercurrent exchange mechanism. And then another concept is this idea of gigantothermy, that large body size allows higher body temperature because of smaller surface area to volume ratios. So now what the heck am I talking about here? Okay, if you were to take a cube like this, right? Okay. You know me, I can't draw very well. I try, but I don't. Okay, so that's a pretty bad looking cube. But now let's take a bigger cube, right? Okay, and we're gonna, let's see, we'll go like this, and like that, and then we'll go like there. Is that a bigger cube? Anyway, I can't draw. You'd think I'd be able to draw by now, but, I don't know. Anyway, something like that. You get the idea. Okay, so let's just say that this cube is 5 on a side, and this cube is 10 on a side. This cube is clearly bigger than that cube. Okay? So what's the surface area of this cube? So let's look at we've got our length dimension here versus our surface area versus our volume. So the length is 5, and the length is 10. So what's our surface area for a cube? That's volume. So 5 times 5 is a surface area, which is 25. But how many surface areas do you have in a cube? 6. So 6 times 25 is 150, right? Is that right? So your surface area is 150. Okay? So now what's your surface area for this guy? Just do the same thing. Oh, no. You're thinking volume again. So 10 times 10 is 100 times 6 is 600. Right? So in this case, you have to multiply 5 times 5 times 6 right? And that's going to equal 150 because that's 25 times 6, which is 150. And in this case, you're multiplying 10 times 10 times 6, and that's going to equal 600. So that's your surface area, right? Okay, so now let's look at volume. Volume is like what you said, length times width times height. So 5 times 5 is 25 times 5 is 125, right? So now its volume is 125, right? And when we do this, it's 10 times 10 times 10 for a cube, right? So that's 1,000. So now let's look at what our surface area to volume ratio is. So now we have 150 divided by 125, and since that equals 6/5, that's going to equal 1.1/5, 1.20. That's our surface area to volume ratio. Okay? But now if we look at this guy, our surface area to volume ratio is 600 over 1,000, and that actually equals, you just move it 3 decimal places, 0.6. Who has the larger surface area to volume ratio, the small cube or the large cube? Small cube. How do you generate heat? Through your volume or through your surface area? Wrong. You generate heat through the volume of you, right? It's your tissue that's undergoing metabolic processes that is inefficient that generates heat. So you generate heat through the volume of your body, okay? But then how do you lose it to the outside world? Through your surface area. So an organism with a smaller surface area to volume ratio is going to lose less heat in relative terms than an organism with a larger surface area to volume ratio. Right? With me? So when you're gigantic, when you're huge, you reduce the surface area to volume ratio. You have more volume to create heat, and you have less surface area through which to lose it. And so therefore, you can increase your body temperature just by becoming large. Make sense? Yeah? Okay. What's that? Well, I don't know if you want to think about skinny people versus bigger people or something like that. Yeah. I didn't say anything, TW. I wasn't looking at you. You know I'm teasing you. But I think it's more when you think about something like a different scale, like orders of magnitude. So if you think about like a small fish versus an extremely large fish, a fish that weighs 10 kilos versus a fish that weighs 600 kilos or something like that. Then you get a difference, and that's what they mean by gigantothermy, right? You become really large, okay, and that makes a big difference. So check out page 95. Okay, but in addition to gigantothermy, you can have regional heterothermy, okay? And why is this important? Well, what happens is in regional heterothermy is you have a counter-current exchange mechanism again. In this case, remember, your tissues are creating heat, right? But once your tissues create heat, they're going to warm up the blood next to those tissues. But then that blood is deoxygenated, right? And so you've got to take that deoxygenated blood and get it back to somewhere to oxygenate it, right? Where do you get it oxygenated? Through the gills. And what are the gills in direct contact with? Cold water, right? So how do you then keep that heat in your body if you're going to give the blood is going to go to your gills and then all your heat's going to go out into the water, right? Because we just mentioned that water carries heat very well, right? 24 times better than air or whatever. Okay? Well, what happens is they have a counter-current exchange mechanism in the vessels that run in their muscles. That is, they're running the arteries right next to the veins. And so when the veins are coming away from the muscles, they're carrying warm, deoxygenated blood, but they're running right next to an artery that carries oxygenated cold blood. And so where does the heat go? The heat goes from the veins into the artery, warming the artery blood before it gets to the muscles. So by the time it gets to the muscles, you have warm, oxygenated blood. And if you do that, you can keep the muscles at a warmer temperature. And here this shows you how fish muscle 47 to 77 degrees or so. I'm sorry, it's so blurry. And in a cold-blooded fish, they're the temperature of the water. Where in this case, a lamnid shark, this would be like a salmon shark or even a great white. They basically have muscles that are warmer than the surrounding.
So basically that explains why they have such bursts of speed.
Exactly. And let's take a look at what that really does then, because they're not the only fish that does that. Tuna also do that. Tuna have regional heterothermy. And here's this example of this countercurrent exchange mechanism. And that countercurrent exchange mechanism is often called a rete, and sometimes it's called a rete mirabile, which means like wonderful net. So here, this rete region is where the blood vessels are running right next to one another, right? And so that warm deoxygenated blood is running right next to cold oxygenated arterial blood, and heat gets transferred from the venous blood into the artery blood before it gets to the muscles. And as a consequence now, the muscles can be a lot warmer. So here's a sort of a temperature isopleth cut through the cross-section of a tuna. And you can see where the muscles run right along, remember right along the ribs, you have the epaxial and hypaxial muscles. You can see that the temperature is much higher than when you get to the outside, as much as about 10 degrees, where the water is 19 degrees, but it's substantially warmer inside the fish's body. Make sense? Everybody with me? And what does that do? Well, if you look at reaction rates of chemicals relative to temperature, you look at activity indices, you look at swimming speeds, and Q10 is another measure of a reaction rate. It's like the ratio of a reaction at one temperature compared to the ratio of a reaction at another temperature. When I mean ratio, I mean product relative to reactant at a 10-degree change. That's what Q10 is. And so you can see that Q10 remains constant but maxed out at temperatures between 20 and 30 degrees or so, that your swimming speeds are heightened at about 30 degrees, that your activity indices at around 20 degrees, and that reaction rates of chemicals, and in this case what I'm suggesting is that proteins that catalyze reactions are higher at 35 to 40 degrees. So as you increase temperature, you increase metabolic processes. And that's going to make your muscles faster, your muscles stronger, etc. Right? Kind of cool, right? And how was this found? So this is kind of cool. A guy came to talk here. He was a friend of Dr. Marv Bernstein before Dr. Bernstein left the biology department. His name was Dr. Fry. I forget his first name. But he found this out. And you know how he did it? He was on a vessel, right? And they were working out in the ocean. And they were fishing. Of course, they'd get their fish sometimes and eat fish for dinner and whatnot. He was on a research vessel. And he pulls up a tuna. Right? And I don't know if you've ever, I've caught bonita before, and you can hold these bonita and their tails just go like, I mean, it's unbelievable how fast their tail can move. And so he had this tuna, and this tuna's thrashing around, and he had a meat thermometer right there. And he grabbed the meat thermometer, and they were actually using the meat thermometer to measure temperatures of water that they were bringing up. They'd drop something down into the ocean, they'd capture some water, they'd bring it up right away and then measure its temperature. And so then he did the same thing with a fish. He just took the meat thermometer on a live fish and stuck it into the fish. And lo and behold, he was like, holy cow, this fish is warm. What's going on here? And so that started a whole line of research for him. And he started studying how do fish keep their bodies warm when they live in these really cold waters. And he studied this whole thing. He came up with this countercurrent exchange mechanism. He deciphered it. And then he went off on this whole thing looking at evolution of counter exchange mechanisms in fishes, because not all fishes have them, but some fishes do, and some fishes that have them aren't related to other fishes, like sharks aren't related. Lamnid sharks aren't necessarily closely related to tuna, right? But they both have these kind of mechanisms in them, right? So pretty cool. So this spawned a whole research program from him, and he studied this for years. So pretty neat, just because he was curious. So serendipity in science. Right? Okay, so pretty neat stuff that these guys go through. And it's not, I'm sorry, it wasn't Fry, it was Graham, Jeff Graham. I have a note to myself. I'm thinking something else. I'm thinking isotopes instead of fishes. Anyway, Jeff Graham was the guy. So sailfish actually have reti in their brains and their eyes to enable their eyes to function better so that they have more acute eyesight, right? So it warms up the tissue around their eyes, right? Which is kind of cool. So you see this counter current exchange mechanism in a variety of different things. I'm going to explain one more counter current exchange mechanism, but before we do that, I wanted just to mention something about tuna in general. Here you see a tuna, large tuna. I guess this woman's maybe, I don't think she's in an aquarium, but she's probably out in the open ocean. And this is, some guy who caught a tuna. That'd be a blast trying to pull one of those guys in, right? But when they fish for tuna, so this is, these numbers represent like mid-1990s, so I'm not even sure what the numbers are now. But if a fisherman goes out and gets tuna with nets, right? A big tuna is worth $6,000 to a fisherman, just one tuna. But when you sell that on the Japanese sushi market, it goes, a 300 to 600 pound fish goes for high quality sushi is $100 a pound. So that fish on the Japanese sushi market is worth $30,000 to $60,000 for one fish.